In this video, we will discuss about bacteria in pancreatic cancer and non-small lung cancer. So, so far we have studied bacteria in the large intestine and in the stomach in our previous video. Here we will discuss the bacteria that are found to be upregulated in the PDAC and the non-small cell lung cancer. So we'll start with this PDAC or pancreatic ductal adenocarcinoma. Previously, it is considered to be sterile uh, conditions, but in the recent studies they have found out that some of the bacteria are upregulated, especially in the short-term survivors of the patients who are affected with PDAC. So this bacteriorodes and Clostridia, these two bacteria are found to be upregulated and is correlated with the upregulation of the myeloid-derived stem cells, T regulatory cells, and the downregulation of the CD8 plus T cells. These conditions are found out in the short-term survivors pancreatic cancer patients, that these two bacteria are upregulated. That might be causing or affecting this pancreatic tumors aggressiveness, but that connections is not yet been established, or there is no general consensus has yet to be established among the researchers, because this pancreatic cancers is very aggressive cancers and it's difficult. People are finding it difficult to identify these pancreatic cancers at the early stage. That's why its survival rate is very low, around 5%. So now let us talk some information about the bacteria in the non-small cell lung cancer. So in this case, what the researchers have found out, there is upregulation of the Streptococcus prevotella, Violinella, and C. pneumoniae, and these bacteria in the microbiota of this non-small cell lung cancer, these bacteria are found to be upregulated. And people have also found out the other things that is caused by this bacteria. One of the things that is important for the tumor survivors that is caused by this bacteria is upregulation of the pathway, that is the survival pathway, which is triggered through P13K, that PDK1, AKT, and this will cause the survival of the tumor cells. At the same time, it will also upregulates this interleukin 1 beta and interleukin 23 through this bacterial antigen. Bacterial antigen will go inside the extravasations through the lungs into the cells, where it will interact with these TL receptors, and there it will cause the upregulations of the interleukin 1 beta and interleukin 23. And at the same time, what will happen is that it will also upregulates these gamma delta T cells and interleukin 17 and Th17 cells, and this will help in the chronic inflammations, and these chronic inflammations will further promote the tumorigenesis in this non-small cell lung cancers. So here the things are quite uh, somewhat clear compared to the case of pancreatic cancers, because here the relationship is not yet been clearly identified, whereas in the case of lung cancers some things are identified, that the upregulations of this bacteria will cause upregulations of the survival pathways in the tumors. At the same time, it will cause upregulations of the cytokines that will cause the chronic inflammations. So this is what has been identified in terms of the bacteria in the pancreatic cancers and the non-small lung cancers. And in this, the CAF refers to the cancer-associated fibroblast. This is also formed, but it is not yet been established whether the bacteria has a role in it, but it has the compositions that is rich of fibronectin and collagen, and that helps in the immune evasions, that by preventing the immune cells, or by preventing the immune antigens of the tumor cells to come in contact with the immune cells, by preventing the tumors antigens that are present on the tumor cells from coming in contact with the immune cells, or in other term we can say that It helps in this masking of the tumor antigens from the immune systems of the host or of the patients. So these are some of the things, or pancreatic cancers and non-small lung cancers related to the bacteria, how some of the bacteria are upregulated and promote the tumors in these cancer cells. If you need to understand more about their uh, relationships in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.